last video I asked you a question: how come the E. coli in our body they are beneficial to us because they make vitamin for us? And how come E. coli outbreak occur in the supermarket? The food company, they have to retrieve their food product because E. coli is harmful to us. How come we have two opposite story? The reason is that it's just like human being. We have different types of human being. Similar, E. coli, they are bacteria. They also have different strain of the E. coli. Certain strain of E. coli that living in our body, they are good one. The E. coli outbreak that occur in the supermarket, they are the bad one. That can cause us sick. Same as some people they are gangster. They will kill you. They will harm you. Some people they are cops. Theoretically, cops are supposed to help us, not kill us. So just like human being, we have different type of human being. We also have different types of different strain of the E. coli. So that's the answer to the question from last video. Again, let me repeat a little bit. There are three structures in the plant cell that animal cells do not have. They are central vacuoles, cell wall, and chloroplast. These three are the structure in the plant cell that are unique to the plant cell. The rest of them, we all have it in the animal cell. Mitochondria, we have it to make the energy. Microtubule actin filament, they are the cytoskeleton. We also have plasma membrane. We have nucleus. We have nucleolus. We have nuclear envelope. We have everything about nucleus. The same thing. The ribosome attached to the rough ER or freely floating in the cytoplasm. We have centrosome. Of course, we have centrosome. Oh, okay. Oh, they have rough ER and smooth ER. They have peroxisome. They have peroxisome too. So previously I said that animal cell, plant cells do not have peroxisome and centrosome, so it is not true. Let me refer to my notes again. I remember there is something that animal cells are unique that plant cells do not have. So plant cells also have peroxisome and centrosome. We have rough ER, smooth ER. You have Golgi apparatus. We have cytoplasm. So this is the electron micrograph of a plant cell. You see all these big green ones? These are the chloroplasts. In the Bio 3B lab, you will see the elodia. Under the light microscope, you will see the chloroplast floating inside a plant cell. It looks very impressive. All right, let's talk about first another organelles. We talk about the cell membrane. Now we talk about the nucleus. Nucleus has a diameter of approximately 5 micrometer. No, you do not need to memorize this number. You do not need to memorize the number. But you need to memorize the function. Nucleus stores the DNA. DNA determines the characteristic and metabolism of the cell. It determines everything. We will dedicate several lessons to talk about DNA. And it contains chromatin. Okay, this is something that we'll talk about. DNA, chromatin, and chromosome. So why do I put it like this, different level? Because DNA is like different level of organization of organism. Remember we learned about from the first lesson. Okay, all animals made of cell, and then the same type of cell they form tissues. Different type of tissue they form an organ. Different organ they come together to form organ system, and then organ system they become living organisms. Similarly, DNA is like the primary structure. When DNA coil together, they form chromatin. when chromatin coil together, further coiling, they form chromosome. So it is primary structure, secondary structure, tertiary structure. Inside the nucleus, we have nucleoplasm. Remember, you have cytoplasm. Cyto meaning the cell. Plasm is like the fluid or the medium inside the cell. We call it cytoplasm. Nucleoplasm meaning that it is the fluid inside the nucleus, and we call it the nucleoplasm. Nucleolus is the rRNA is made. Ribosomal RNA. Right now, we have not talked about what rRNA is. We have not talked about the function of ribosomal RNA, but it is very important in protein synthesis. We'll talk about it in much more detail later. You will need to know this, but right now just memorize it. Ribosomal RNA is made in nucleolus. Nuclear envelope, it has another name, nuclear membrane. So it is a double membrane that separates the nucleus from the cytoplasm. Remember, cell membrane or plasma membrane is a phospholipid bilayer. So it has two layers. Similarly, nuclear envelope, it is a double membrane that surrounds the DNA. And it separates the DNA from the cytoplasm. Now you can imagine, wow, how important the DNA is. Because DNA is surrounded by the double membrane of nuclear envelope, and then outside is surrounded by phospholipid bilayer. So it is surrounded by 4 layers of a membrane. And then nuclear pores. The nuclear envelope, although it is double layer, it has a nuclear pore. That means it has holes on the nuclear envelope to allow the movement of, let me give you a preview, something called messenger RNA. Messenger RNA can go from inside the nucleus to go to the cytoplasm. So this is how it looks like. The purple ball is the nucleus. And then when we enlarge it, you can see that on the nuclear membrane here, you have this nuclear pore. We form a hole here. This, it is a double layer of the membrane. Inside the nucleus, you have this nucleolus here. This is how it looks like under the electron microscope. Ribosome, the function, you have to memorize it, is the site of protein synthesis. It uses messenger RNA. I told you that messenger RNA goes from inside the nucleus to go to the cytoplasm. This is what I call messenger RNA. So when the messenger RNA going from the nucleus to the cytoplasm, then you will go into the ribosome. Messenger RNA will go to ribosome, and then you will start to make protein. And then ribosome has 2 subunits, large subunit and small subunit. Yes, they literally call it large and small. The ribosome is made up of what? Ribosome is made up of rRNA, ribosomal RNA. Where did we see it before? Remember, we see it here. So nucleolus make ribosomal RNA. And after you make rRNA will move from the nucleus to the cytoplasm through the nuclear pore, and then rRNA will become the ribosome. They will group together with other protein, and then they will form the ribosome, the large and small subunit. Now it makes sense. That's why the nucleus is very important. A lot of things are made inside the nucleus. And then we can also find ribosome. There are 2 places we can find ribosome. One is that in the cytoplasm. In the cytoplasm, they will group together to form polyribosome. So they're freely floating in the cytoplasm. And most importantly, you can find ribosome on the rough endoplasmic reticulum, or we simply call it rough ER. Rough ER is another organelle that is found right next to the nucleus.